the fluid in your body can be divided into the fluid inside of cells versus the fluid outside. The fluid outside of cells can all be traced back to the plasma, because blood pressure is constantly pushing plasma across capillary walls, constantly. Depending on where the fluid is, once it reaches the other side of the capillary wall, it has a unique name and function. If it's in the ventricles of the brain, we have capillaries in the ventricles of the brain, so once we push the plasma across, if it's now in the ventricle, we call it cerebrospinal fluid. We have capillaries in our synovial membranes. You push the plasma across, it ends up in the joint cavity, we call it synovial fluid. Capillaries in the eyes, forming the humors of the eye, in the inner ear, the endolymph and perilymph. So these are all compartments of ECF. Even the plasma is in its own compartment, right? Plasma refers to the fluid inside of blood vessels and the heart. Now, if we're pushing plasma across the capillary wall and the capillary is just in a loose connective tissue, once it reaches the other side, it's not in a particular compartment. It's just filling up that space between cells within the tissue. It's called interstitial fluid. These two combined account for 90-something% of all ECF. These are the two big subdivisions. Now, we're constantly making interstitial fluid, which means we constantly have to remove it at the same rate, otherwise we get swelling. Lymphatics are responsible for picking up the excess interstitial fluid, which we could also say is excess capillary filtrate from the plasma. But then eventually, if you follow the path of lymph, those lymphatics end up emptying into veins and returning the lymph back to the blood vessels, so it becomes plasma once again. So this is just a cycle. But this is also a reminder why we don't have to monitor all of these subdivisions separately and make adjustments to them separately. We just monitor plasma and make adjustments to plasma, and that takes care of everybody else.
What was CSF again?
Cerebrospinal fluid. So you'll learn about synovial fluid in Chapter 8, CSF in Chapter 11, lymph is next semester, humors of the eye at the end of this semester. Now, were there some other fluids, some other name, like ground substance?
Yeah, ground substance is the same as
interstitial
fluid.
The three main components of any connective tissue: cells, fibers, ground substance. Although you can take the ground substance and the fibers, and that's your ECM, right? So really, connective tissue is cells, and then everything outside of them, the extracellular matrix. And so the ground substance is interstitial fluid. Mm-hmm. Talking about the same stuff. We connected this idea to blood just to show why blood is considered connective tissue. You have the blood cells, which are the formed elements. You have the fluid component, plasma. You don't have fibers per se unless the blood is actually clotted. Otherwise, you just have plasma proteins called fibrinogen. And through the clotting reaction, it gets converted into fiber. So if blood is clotted, you clearly see the three components of connective tissue: the cells, the fibers, and the ground substance. Were there any others? That pretty much cover it. We talked about serous fluid, which is yet another extracellular fluid. And in a very general sense, that just means a watery solution, a watery fluid. Right? So we can even classify exocrine glands as serous, mucus, or seromucus.
I guess one question would be, so the ICF is always in the cell versus the ECF is always outside, but at what point, how would you know if it's ECF or if it's interstitial fluid? Is there a way
These
to-
are all types of ECF.
So you could say it could be ECF or ICF if it's outside of the cell?
Did you say ICF or ISF?
ISF, I'm sorry, interstitial
If,
fluid.
okay, so
Yeah.
the fluid that's outside of cells but
Mhm.
not in its own confined compartment, it's interstitial fluid. If it's in a compartment, it gets a different name. So in lymphatics, it's lymph. In a
Right.
synovial joint, synovial fluid. In the eye, humors, right? They're all in a compartment, a confined
You know, when
space.
you have all those little cell pictures on the histology, is that then always ECF?
ISF.
ISF?
Yeah.
Okay,
We're always going to be specific. We're
okay.
rarely just going to call something ECF because it's too broad. That means anything
That means any of that.
that's not in a cell, any fluid that's not in a cell. So we're going to use one of the more specific terms.
Okay.
You got me for 25 more minutes. What's next? I'm sorry, I just hear the fan above me.
Can we talk about the control of cell division for tissue?
So like the cancer stuff, the terminology related to cancer, or is this a separate topic?
No, this is the one that's right before the cancer issue. This one's the aspect of tissue,
Mm.
control of cell division,
Okay.
differentiation, aging.
Chapter 4. Yeah, that should be chapter 4. Show me. Oh, chapter. Chapter. So objective number 5
On chapter.
for chapter 4. So that's getting towards the end. So if you go to the very end of the study guide, right before the APS manual questions, developmental aspects of tissues, Roman numeral 12. Mitosis requires a lot of energy, ATP. This is why starvation leads to poorer growth if you're a child and you're still growing, or as an adult, starvation leads to poor healing and maintenance. Because as an adult, there doesn't have to be damage for us to need to produce more cells and more tissue. There's also just the turnover. The fact that some of these tissues don't last very long. Epithelial tissue in particular, especially where it lines the gut, doesn't even last a week. So those cells are turned over at a really high rate. And that's expensive. It's going to cost a lot of ATP and a lot of raw materials, right, to constantly be making cells. So you have to have adequate nutrition. Also related to development and growth. The idea that we start with one cell, the zygote, right, from which this multicellular organism is produced, which is what we are. We consist of 75 trillion or so cells. Yet they all have the same DNA, right? And so the way we can have different cell types that look different and carry out different functions is simply by having different cells activate different genes. No one cell in your body uses all of the genes that it contains. No cell does that. So it's just a question of what group, what set of genes are going to be activated, and therefore what set of proteins is this cell going to make. And that's what determines its structure and its function. And that process of activating genes and creating a unique specialized cell, that's differentiation. Right? So that zygote doesn't have any particular function to carry out other than divide, divide, divide. right? That one cell divides into 2, those into 4, 8, 16, 32, 64, 128, 56, on and on and on and on. Now, once we have thousands or tens of thousands of cells, and this is early on, this is pre-embryonic stage, then they can start to specialize. Then we start to see groups of cells forming. You know, this group is going to be called the epiblast cells, and this group is the hypoblast cells. And then from there, they differentiate more and more and more. We get more and more different cell types. You don't need to know about any of those names or even that process, just the general idea of differentiation, of how we have these cells that have the exact same DNA, right? Yet they become very different in structure and function. Cells that have a high turnover rate, like epithelial cells, are going to require a lot of energy, a lot of raw materials. So that goes back to starvation and being malnourished and how that affects growth and maintenance. Some cells don't turn over at all. This would be tissues that don't regenerate. Neurons, right? They don't turn over. Cardiomyocytes don't turn over. Those are meant to last a lifetime, so you have to take care of them. And when you have a tissue like this that doesn't regenerate and it's damaged, what's going to replace the damaged tissue? Scar tissue, fibrosis. What are the tissues that regenerate really well? Who's at the top of the list? And? What always goes along with epithelium? They're buddies. Areolar connective tissue. They always go together. And then aside from those two, dense irregular connective tissue, which actually does go along with epithelial and areolar, because those three together form the cutaneous membrane, which is exposed to the external environment. It's going to get damaged. So those three tissues better be able to regenerate well. And besides those three, blood and bone. And then not at all. Which ones do not regenerate at all? Yeah. Cardiac muscle, nervous tissue. EGF? Who's got it? Epidermal growth factor. What does EGF stimulate when we sprinkle it on some epithelial stem cells? Epidermal growth factor. It's going to stimulate growth of an epithelium. Now, if we want to grow an epidermis, this chemical EGF needs to do something other than stimulate growth. Because that'll give us an epithelium, but if it's epidermis that we're after, it needs to be keratinized. So EGF stimulates both mitosis and keratin formation, promotes keratinization. So truly, we're getting an epidermis from it. As far as that objective, did we touch on everything?
Yeah.
The effects of aging, that was FYI. I'm not going to test you on that at all. Just a few little interesting tidbits in the chapter reading. What is apoptosis? Programmed cell death. Programmed meaning there are genes that code for proteins that carry out this particular action. And it's just a matter of activating those genes. And when are cells programmed to die? What sorts of things would lead to the activation of these genes? When they become old and dysfunctional, or
Damaged?
crowding. Yeah, tumor. When they're getting too crowded, too close together. It's called contact inhibition. But when they start kind of pressing against each other, that's going to result in the release of chemical signals that activates the genes that code for the proteins that carry out apoptosis. And then related to that, something I mentioned in passing, but you don't have to worry about it till next semester, some of our immune cells can actually trick cells into activating those genes, into using their own sort of self-destruct mode. T lymphocytes do that. Natural killer cells do that. They release chemicals that can cause an abnormal cell to basically kill itself. Which glands are considered ductless? Endocrine glands. So sorry. You gotta yell at me.
Would you classify sebaceous glands as follow my train of thought?
I heard hair follicle.
Yeah, oh yeah, okay, so would those be the same, even though they use a hair follicle as a
So their name. Oh, are you talking about endocrine exocrine?
Yeah,
They're exocrine glands.
right.
She's asking if they're ductless, they don't have their own duct, so they use a hair.
No, they have a duct.
They do have-
Their duct leads into the hair pore.
Okay.
Apocrine sweat glands also have a duct that leads into the hair pore. So when I say that they don't have their own duct, what I'm really saying is they don't have a duct that goes all the way to the surface.
Right, and that's just apocrine,
Eccrine has
right?
its own duct that goes to the surface. Apocrine and sebaceous do not. Who produces the glycoproteins of the clear layer of basal lamina? That's what that clear layer is. It's a layer of glycoproteins, but who is secreting them? Who makes that layer? The epithelial cells. Who makes the fibers of the reticular lamina? Fibroblasts, the connective tissue cells. So the basement membrane is partly made by the epithelium above it, partly made by the connective tissue cells below it. It's like a shared structure. What layers of the epidermis consist of dead, flat keratinocytes? Stratum lucidum and stratum corneum. What makes the epidermis water resistant? Glycolipids around and between the keratinocytes. And what makes it hard for water to actually get through the cells?
Keratin.
Right? So hard to get through the cells because of the keratin, hard to get between the cells because of the glycolipids. What is coating the surface of the epidermis that also contributes to water resistance? Take oil from sebum.
That's what I was going to say, but I thought, but what about not oil free? And she water resistant.
Oh, there you go. No, no, no.
So for that, going back to that question, that same rule doesn't apply though to thick skin.
What? Sorry, which
The
rule?
oil that's on the surface of the skin,
Right.
that adds to the water resistance, that doesn't apply to thick skin. So
Sure.
for thick skin, it's just going to be the glycolipids between the cells, the keratinocytes, and the keratin. That
For thick
keratinocytes
skin,
are made
yes,
of, right? Okay.
correct. But what else? What is it about thick skin that makes it even more water resistant than thin, even though it doesn't have the sebum?
The thickness.
The thickness of the cornea.
So what, if we don't have sebaceous glands, if we don't have apocrine glands, what is it that leaves behind your thumbprint?
There's always.
Some kind
I know we call it, say there's oil on our fingers. It's not like literally oil from like a sebaceous gland
Okay.
necessarily. I think it's more a combination of just kind of dirt, picking up things that we touch, the moisture on it, kind of collect stuff and
cells left behind?
yeah, just a little bit of everything. And I'm sure there's some oil there too that we just pick up from somewhere, but it's not like it's secreted onto it. You know, if you washed your hands really, really, really good and then touched a piece of glass or something, the fingerprint left behind wouldn't be nearly as noticeable as if your hands were dirty, right? And you actually had, you know, dirt and oil and all kinds of stuff on your hands. But I know what you're saying.
What's a thumbprint behind?
Ah. Genes that code for normal processes involved in growth, membrane junctions, mitosis, genes that are involved in mitosis, growth, formation of membrane junctions. What do we call those? When they're still normal. Because those are processes involved in cancer if those processes don't go right. If those processes fail in some way. Proto-oncogenes.
So it's proto-oncogene.
Proto.
Not proto.
Proteo means protein.
Okay.
Proto-oncogenes. So normal processes, right? But when those normal processes don't get carried out in the right way, it can lead to cancer formation. So what happens if a proto-oncogene becomes mutated?
It becomes an oncogene.
Now it's an oncogene. What does oncogene mean?
Cancer cell.
Cancer-causing gene.
Okay, yeah.
How about genes that code for proteins that are involved in limiting growth,
Oh, it's just one.
preventing cancer formation, essentially? Anti-oncogenes or repressor genes, or what does TSG stand for? Tumor suppressor genes. What's the most common TSG involved in at least half of all cancers? TP53. What is the term for new growth?
Neoplasm.
If the new growth is confined within an epithelium or connective tissue membrane barrier, we consider it to be benign. If it breaks through the barrier, cancer. Right? And cancer cells are aggressive and they break through basement membranes and connective tissues, and they can get into the blood and lymph. And if they do that and they start circulating around the body, what has happened? Metastasis. What's the most dangerous type of skin cancer? Malignant melanoma. In what layer do those cancer cells arise? Stratum basal, because that's where you find the melanocytes. Basal cell carcinoma. Where do those cells originate? Stratum basal. Squamous cell carcinoma. In what layer do those cells originate? Given the name squamous cell, you would think lucidum, corneum, because that's where they're really, really flat. But those are dead cell layers, so no cancer comes from dead cells.
Is it spinosum?
It is spinosum. They're dying in granulosum. They're still living cells in spinosum. So yeah, the cancers are coming from basal or spinosum. What do you call a substance or just anything that is cancer causing? Carcinogen. Which membrane does not form a barrier between internal and external environments? Serous. The two that do separate internal and external, mucus and cutaneous, which means the only way in or out of the internal environment is through mucus or cutaneous membranes. What pigment is converted into vitamin A and gives an orange color?
Keratin.
Why do the cells of a stratified squamous epithelium die?
A blood supply.
Too far away from the blood supply. Right? So the upper layers. Modified sweat glands, 2 types. What are they called?
Mammary and
Starts with a C.
ceruminous.
The name of the secretion for each. Cerumen, layman's term, earwax. It's called cerumen.
And milk.
And milk. And the secretion from sebaceous glands. Sebum. And the secretion from the apocrine and eccrine sweat glands. Sweat. Characteristics of cerumen.
Sticky, a little oily.
Sticky and
Bitter.
So most likely the function is?
Keep insects out.
Keep the critters out. What may form on the skin if you have excess scar tissue formation? Keloid. What's it called when your hands, usually fingertips, palms, are constantly being abraded by doing some physical labor and it thickens? Callus. Where do you find stratified cuboidal epithelium? Ducts of some large glands. All right, give me 2 large glands. Sweat glands, yes. So we're talking about glands that have a duct. So apocrine and eccrine sweat glands, they both have ducts, right? It's just that one duct leads into a hair pore. The other duct actually goes all the way to the surface. And then mammary glands. Sweat glands and mammary glands would be 2 locations for stratified cuboidal. Function of stratified cuboidal. Ooh, this is a good question. We haven't asked this one. So anytime the epithelium is stratified, you can say protection, right? Always. But this is lining a duct, so it's not just, not lining, it's forming the duct, so it's not just protection, it's also secretion. So it's not just secretory cells that secrete into the lumen, duct cells also secrete into the lumen. Protection and secretion for stratified cuboidal. And if stratified columnar is doing the same thing, forming the duct of a large gland, you would again say protection and secretion. Simple cuboidal. Typically, what's the function? Secretion and absorption. Kidney tubules, reabsorption. Locations where you find simple cuboidal other than kidney tubules. Right, so if stratified cuboidal is ducts of large glands, simple cuboidal, ducts of small glands. And covers the surface of what organ? Ovaries. Immediate event occurring once a damage has happened to the skin, a cut, a scrape. Immediate response, immediate events. Blood clotting and inflammation. Those 2 things are immediate. Hours later, what happens? Assuming it can form? Granulation tissue will form. What's it made out of? Fibers from the blood clot. And fibroblasts, those are going to be your stem cells, and capillaries. So the fibrin mesh provides a scaffold for the stem cells and capillaries to migrate into. And then the cells can start secreting the new matrix. capillaries, of course, provide the nutrients. Cool. We went over everything on the exam. Just in time. 30.